As Nick's alluded to, we've done a lot of work together as a working group, carefully pre-processing all of the raw accelerometry information to get out some good stable measures of physical activity intensity, and that's taken a lot of careful work. And what I'm going to describe over the next 10 minutes is additional work that we're doing to try and further enhance the Biobank resource for you and other researchers around the world who use this fabulous resource. So Nick's already introduced various work done by other researchers showing fabulous uses of the resource. Another example I quite like was done by Danny Smith and his group up in Glasgow, using the derived measures we put back into the resource to try and get at measures of circadian rhythmicity and showing some nice strong cross-sectional associations of mood disorders as well. So this is a resource that's been used to try and answer questions for many different disease outcomes as well, and exposure types. And the work I'm going to talk about is work that we've done to try and get at more functional or behavioural measures from the accelerometry data, such as trying to identify when and how often individuals might be sitting or walking or driving or sleeping, and how we try and achieve this is via statistical machine learning, essentially a supervised machine learning exercise from the raw accelerometry data trace that you can see on your left. Is there any patterns or signatures inherent in this that can help us identify behaviour states of interest? And there have been some past efforts of this, but there have been two fundamental flaws. Firstly, on the technical point of view, the choice of models has often been inappropriate for the type of data. And secondly, in terms of the training and validation of these machine learned algorithms, it's taken place in unnaturalistic laboratory environments. What we'd really like to know is what participants are doing in their everyday free living lives. So to achieve that in Oxford, we asked 150 participants to wear a camera-based device as they went about their everyday activities. And we weren't so interested in trying to get a representative set of people. What's more important is getting a representative and diverse set of behaviours in which to train the model. So the camera-based device looks a little bit like this that people wear via neck-worn lanyard, and the data then we get back looks, as you can see in the video here behind me, whereby from the camera data, so it takes a first-person point of view image every 10 to 15 seconds, and we can see quite clearly whether a person is walking or whether they're sitting or whether they're driving. We can even capture other lifestyle health behaviours, such as whether they're having a sneaky fry-up particular parts of the day as well. And then our challenge and interest in this from a biobank perspective is all these participants had the same accelerometers in the UK Biobank, and we extract out a series of time and frequency domain features. These ones are orientation invariant. We run those through what we call a random forest classifier to make some preliminary predictions on the type of activity the person is doing, whether it's sleeping, walking, running. And because of the time series nature of this data, we can apply a second machine learned approach, namely hidden Markov models then, to smooth over those predictions to further improve the accuracy of the classifier we've done. So this is work done in collaboration with Chris Holmes, who's one of the founding directors at the Alan Turing Institute. And what we had then, sort of in this particular paper published below, is from 130 people. We had 160,000 minutes of annotated human behaviour in free living scenarios. It's around 15 to 20 times larger than any laboratory type data set, and the key thing is the environment in which the data is collected. It's not a constrained laboratory environment. The overall accuracy then to identify these different types of activity then was 87%, with particularly getting good success in terms of distinguishing between sleep and sitting behaviours, and we can also identify activities such as walking quite well too. So that's one form of validity around this machine learning approach we've taken. We've also then applied this model to the Biobank participants, so all 100,000 UK Biobank participants. And we wanted to first of all see whether there's any face validity to this. So does the output of this classifier make intuitive sense? So we've got two different plots to show you here. On the left-hand side, we've got accelerometer predicted time spent asleep. So X x-axis is hour of day, y-axis is probability of a population group being asleep. And we've got two different groups been shown here. Those who self-report themselves as being definitely a morning person versus those who self-report themselves as being definitely an evening person. And we're getting very distinct differences, for example, at 8 a.m. in the expected direction. And then over at the right-hand side, we've got what I like to call the Batman plot, whereby we've got accelerometry predicted bicycling time, whereby we've split up the groups by those who self-report themselves as being cycle commuters versus those who commute to work via other means. And again, we're getting big spikes in cycling-related behaviour at key commute times. So this gives us further confidence that these machine-learned models have got a face validity as well as the validity in our detailed ground truth data set in Oxford participants. So that has all helped us, as I said, on the 100,000 UK Biobank participants. Wants to do some QC. This particular plot is in 91,000 participants, whereby we can build up this 24-hour model of human movement behaviours as identified via statistical machine learning. And of course, the beauty of UK Biobank is that all these participants got a range of other variables as well, including the imputed genetic data. So working along with Cecilia Lindgren and her team at the Big Data Institute in Oxford too, we've been able to identify variants then associated with, for example, sedentary behaviour status and also loci identified with sleep duration. And as we would have seen just after the lunch in the afternoon, Sam Jones from Exeter and Mike Whedon and their team have done some very nice complementary work as well on trying to get out other measures of sleep-related phenotypes. And we're getting very similar stories in both forms of analysis, that some of the sleep duration loci overlap with our prior knowledge from self-reported GWAS of sleep, but we're also identifying new loci as well. And indeed, looking at the overall activity trait within the UK Biobank, we've got here is a Miami plot. Think of it as two Manhattan plots. So in the bottom, we've got a genome-wide association study against self-reported physical activity status in 351,000 participants. On the top side, then, we've got a genome-wide association study on the accelerometry-derived overall activity trait in 91,000 participants. So unsurprisingly, we still identify more loci using the self-reported metric because we had a much greater sample size. However, with the objective trait in the top, we're getting much higher estimates of heritability and also very clear signals in chromosomes 10 and 17 that we hadn't seen before using the self-reported data. So again, I think this is a wonderful example of having subjective and objective measures at scale, and this is again where UK Biobank is just truly unique in the world. And these loci we identified then allow us to understand more around the etiology of movement-related behaviours. For example, partitioned heritability is showing that the central nervous system is playing an important role, quite expected given its behavioural traits that we're dealing with. And furthermore, the loci were identified are proving to be, I think, better than we would have originally expected as instrument variables for Mendelian randomization analysis to try and unpick potential routes of causality. So, for example, our preliminary MR is suggesting that increases in overall activity are associated with a reduction in diastolic blood pressure and also odds of hypertension. And conversely, that increases in sleep duration are associated with an increased odds of hypertension. And more careful follow-up work will need to be done in this, particularly in terms of replicating in other cohorts as well. But again, I think it opens up very exciting opportunities to further our understanding of the causes and consequences of physical activity and sleep duration type behaviours. So to summarise, as Nick has alluded to, these metrics that we've been working together in a working group for the last few years are all available and already used by researchers in the Biobank resource. The machine learned phenotypes and sleep duration, sedentary behaviour and walking that I've just presented, they will be returned back into the resource, so it'll hopefully then further enhance it for other researchers to use for their particular research questions of interest. And of course, there's many exciting possibilities to further enhance the resource, where, for example, the Alan Turing Institute have given us some funding to look at unsupervised learning and to see other new health-relevant patterns of activity that we might not have thought of before. So with that, I'll stop and thank you very much for your attention.